And we are online. So welcome to everybody. This will be our last webinar for this season, the third season of the International Legume Society webinars. I'm very happy to be here with Dr. Janine Croser from SARDI, Crop and Pasture Improvement Lead at SARDI. And today we are going to talk about rapid allele mining, which is an interesting and topic with growing attention on it. So please, Janine, the mic is yours, and thanks for your
Nelson.
presentation.
Thanks, Nelson, for the kind introduction. And it's nice to see Tom online there. I don't know, he's disappeared again, but I haven't seen him for many years. So hello, Tom, and hello to everyone else online. Today, I'd like to tell a story about the combination of ideas from others, really, and research that we've done over many years, which has led us to a robust way to accelerate generation cycling across species. I'm planning to lead us through the development of the protocol and how we've integrated vertical farming and some phenotyping into a rapid acceleration platform. I'll provide some examples of how we use the platform alongside things like micro-assisted selection for rapid cultivar development across species, and how we've modified the protocol to rapidly mine for beneficial alleles from wild relatives. And I'll use an example from chickpea. And also how we've integrated with dormancy breaking to rapidly breed new pasture species. And in this case, I'll use a strand medic example. I'd also like to take the opportunity to introduce some of our future plans here at SARDI, how we plan to speed the development of double and triple mutants that we've identified in oats using the Carlsberg Laboratory Tratomic reverse genetics approach. And I'll just provide another final example, how we're using it in bread wheat to rapidly bulk seed for field trials. So without further ado, I will move to the next slide, which is actually my thank you slide, because I think it's really important to acknowledge that the work I'm presenting today is a result of the contributions of many people. Firstly, the excellent team that I worked with at UWA, which is the University of Western Australia in Perth. I had the immense privilege of working with this team from about 2015 to 2021 on this topic and a range of related projects. The UWA team helped to determine the fundamentals of the platform that I'm presenting. They helped to value add through the addition of the innovative phenotyping and to deliver our research on an industry scale. Particular shout out to Drs. Maria Pathos-Navarro, Federico Rabota and Richard Bennett, who were research fellows within my team and who lead various components of the research. Maria and my valued colleague Judith Lichtenszweig, who you can see in the picture there, were also instrumental in the work I'll be presenting on wild sisa. And I'd like to give a special shout out to my mentors, Dr. Monica Lulsdorf from University of Saskatchewan, Dr. Sergio Schatt from INRA and Willie Erskine from UWA. Sergio and Monica were also involved in a lot of the generation of ideas for this work. Monica has concurrently developed platforms at USask as well with her team. So it's been very much a group effort. In 2022, I moved to the South Australian Research and Development Institute, and this has broadened the scope of work I'm involved in as I lead the crop and pasture improvement group here. This has provided us opportunities to implement the rapid cycling platform in new and kind of interesting ways. A big thanks to my colleagues here at SARDI, Drs. Judith Atieno, Julie Hayes, John Harris, Yusuf Genk, David Peck, and Victor Sadras, who've generously provided information into this presentation as well into the latter parts of what I'm presenting today. And finally, of course, no work is possible without funding. And for us in Australia, our major funders are the Greens Research and Development Corporation. They are the major investor in much of the work I'll be presenting today, but there has been a range of other valued contributors and their details are provided in logos at the end. So why is this topic important for us to consider as a group of plant scientists? I think I'd like to start that with a personal perspective. I've come from a family farm myself in Victoria, here in Australia. Last season, we managed to cut just a small amount of hay off failed crops, and that was due to a very significant drought. And what the drought did not kill, the hail finished off. And this is in a region normally fairly reliable and highly productive in terms of grain growing. For this year, we've sown our crops and we are waiting for rain still. So in South Australia, here in Adelaide, where I'm based now, we're currently in the grip of the driest 16-month period since rainfall record keeping began here in the early 1900s. For the first time in my life, I'm seeing the dieback of native trees across the hills that surround Adelaide, even our eucalypts, which are normally highly resilient to changes in climate. And at the very same time, there's unprecedented flooding in New South Wales, another of our states. They're having 1 in 500-year flooding, which impacts agriculture across a broad part of another, usually highly productive region in Australia. And the flood levels there are above the previous record set in 1929. So I think as scientists, it's clear we're in an arms race against climate change impacts on our food production systems, and we're really feeling that here in Australia this year. I also like to point out that new regulatory frameworks are coming into play for us here in Australia, and I'm sure for our, our colleagues across the world. For instance, a climate-related financial disclosure regime has just been brought into fruition on January 1st of this year. That's compelling large corporations to start to report their Scope 3 emissions. Now, for many of those large businesses, farms are in their Scope 3 emissions. So that's putting a regulatory burden back on our farmers to really look at carbon accounting, environmental footprinting, those sort of things. So they're going to really need us to step up in terms of genetics to ensure that their kilograms of grain per kilograms of CO2 are maximised. There's also some maximum residue limits for ag chemicals that are starting to come into our regulatory frameworks, and there's a move to global harmonisation with what other peoples, particularly the EU, are doing in that space. So it's clear, I think, that we need to turbocharge the speed at which we identify, access and integrate beneficial alleles into our plant improvement pipelines. And any tools that support that rapid integration are going to be essential for modern breeding programs, aiming not just for productivity, but for resilience and adaptability as we move forward in an increasingly uncertain biological, regulatory and climatic framework. So we need it. Allele mining, the subject of my talk today, focuses on the systematic recovery of the underutilised variation. And in my case, the work that I'm presenting will touch on wild relatives, land races, legacy cultivars, mutant populations, and the reaching back and bringing this diversity back into our cultivated lines provides a buffer against genetic erosion in your elite breeding pools. And we all know that those breeding pools tend to narrow over time. And there's a good example of that in Figure A here. It's also interesting to think about the opportunity to reintroduce phenotypic plasticity to plants. And, by going back into your wild gene pools, that can assist in that process. I think, it's easy for us to agree that the allele mining underpins all of our plant breeding objectives and will be the key to meaningful genetic gain. That is not supposed to be in this particular presentation, so I'll keep going. So what do I mean by genetic gain? In this situation, genetic gain measures how much a breeding program improves a trait or a set of traits within a population through artificial selection. And this is a term originally coined by our animal geneticists. Essentially, it's simplified to be genetic diversity by population size by selection accuracy over generation cycle. And one of the two things that drove our interest in accelerating the breeding cycle was this recognition of how critical breeding cycle time is to genetic gain overall. If we move to the next slide. So obviously our breeding goals are to obtain those high rates of genetic gain for all of our traits of interest while maintaining the genetic diversity that's required to sustain our rate of adoption and use of improved varieties by end users. So it's really important to start thinking keep thinking in those sort of frameworks when we're talking about genetic gain. So what is the rate of genetic gain looking like in some of our key crops? Well, in wheat, it's about 0.9 to 1.2% per year, and the typical breeding cycle for wheat's about 10 to 12 years at the moment. It is reducing as new technologies come into play. If we focus on legumes, we can see a couple of recent papers by Victor Sadras and his team here at SARDI have shown that it's actually, not too bad. In lentil, it's 1.2% per year, and in chickpea, it's not quite as good at 0.6% per year. Now, that's attributed to a focus on disease resistance, on grain quality traits, but it is problematic. And I think we all understand that to meet food security, we need high levels of genetic gain. And the estimate in wheat is that we need about 2% genetic gain per year to meet the global demand that we currently have. Population growth, decreasing arable land, climate change, they're all increasing the demand for food and requiring from us a higher rate of genetic gain. So that led us to the question, how to most effectively accelerate and new allelic introgressions and to drive improvement in the rate of genetic gain for key crops. And our original focus for this work was, in fact, cool season grain legumes. So you'll see a lot of the focus here is on that. It's my new versions appears to have not got rid of the hidden slides, I'm afraid. So if we look at the fact that this is not a new question, plant breeders and pre-breeders have been finding innovative ways to achieve this objective for more than 80 years now. Norman Borlaug, who's widely known as the father of the Green Revolution, was working with dwarfism, nitrogen, and was an early adopter of shuttle breeding to speed generation turnover per year. In the early 60s, Guru Maheshwari pioneered laboratory-based haploid techniques, and that later led to commercially applicable double haploid protocols across a range of important food crops, including your cereals and oilseeds. Unfortunately, these protocols are yet to be applied at commercially relevant scale to any of the crop legume species. Single seed descent was first proposed by Golden in the 1940s and promoted by Brim and others in the 60s. And the approach of taking a single seed forward and growing plants under controlled environment conditions arose from this early work, and it really forms the basis of our approach to accelerate cycle turnover. On a bit of a side note, I'm currently working with a PhD student, and part of their study is using a Monte Carlo modelling approach to assess the point where generation cycling platforms, like I'll be describing today, become more cost-effective than double haploid platforms in canola. So that's just something we can come back to in the questions if anyone's interested in that. So after many years of frustrating research trying to adapt double haploids to pulse crops, we decided to take a fresh look at other ways to accelerate the progression to homozygosity. And this led us back to that single seed descent idea. And if we take the idea of a diploid selfing plant, for instance, chickpea, the initial cross is undertaken, followed by bulking of your F1, and then you return to homozygosity through successive periods of inbreeding, and that's what's seen here in this F2 to F5. There's obviously different points where breeders can choose to make selections while they're moving towards homozygosity, but generally they usually have to be satisfied that it's sufficiently homozygous to prevent phenotypic bias. And when we considered the opportunity for accelerating the breeding pipeline of the crop legumes, it was the speed of going from this F2 to F5 that was the clear target for us for intervention. So we considered the full plant life cycle time, of flowering time and seed maturation, and started to dissect that and to think about, well, how can we speed time to flowering? And then how might we speed time to seed maturation? Now, as we know, and as was discussed sort of previously, SSD has been around for a long time. It relies on a low biomass and a fast rate to reproductive phase. So we already knew from the excellent examples that exist in the literature that photoperiod and temperature are the key determinants of flowering time, and they're the big ticket items. You cannot compress flowering time without photoperiod saturation and without a sort of a spring to late spring type temperature. So that goes without saying. However, what we wanted to know was, could we further improve the compression? Could we further improve synchronization of different phenologies by looking at light quality and using light quality as a way to compress the flowering time differences between, say, early and late field flowering types, and just to try and further reduce the time to flowering overall. So many of us know that plants receive and are able to perceive light signals through different receptors. We were looking at the shade effect and the effect of what's called shade avoidance on compressing flowering times, and there was good evidence in literature that the changing ratios of light quality could in fact trigger shade avoidance, which is telling the plant to hurry up, finish your life cycle before you get fully shaded by whatever it is that's nearby. If we're going back to sort of Devonian period evolution, or more recently before canopy closure in monocultured crop species. So in our quest to reduce this time to flowering, we really had a good look at the early work in Arabidopsis and the effects of enrichment in the far red non-visible light spectrum on accelerating the progression to the reproductive phase. That led us to a hypothesis that this far red light, which we say is 700 or 710 to 800, 850, which is usually transmitted by the plants, is a great way to tell the plants that you're actually about to be shaded, changing the ratio of that light in their spectrum that you provide to them. It provides that environmental signal to the plant that they're in a shading risk situation, and then they basically receive the signal, and it's to do with the amount of carbohydrate that they're receiving due to photosynthetically active radiation, to turn on that shade avoidance response. For Arabidopsis, like I was alluding to in the last slide, a low red to far red ratio has been really clearly shown to accelerate flowering, and we were keen to see whether that would work across our species as well that we were interested in. Conveniently, at the same time, there were recent advances in LED light technology that opened the door to these more affordable, highly controlled spectral manipulation options. LEDs, if you're unaware, they release energy in the form of photons, so you can also use them as sort of photon counting mechanisms. And the LED grow lights that we used were developed by various companies, and the example that I'll show here today is from Valoja in Finland, and we've used their LED technology extensively in our work. They provide these different arrays, and you can see on their website that they have various different forms of light spectral outputs from these arrays. So we selected these AP67 arrays and AP673 arrays, which were enriched in that far red spectrum, and undertook a series of experiments to better understand the effect of light quality on time to flower in the long day cool season legumes. It comes as no surprise that as soon as you extend the photo period, you get an extension to, not an extension, a compression to your flowering time. And that's what we're seeing here across lentil pea, chickpea, faba bean, and lupin. So we've got a natural day length. And then in the red, what we have is this AP67 array, which has got a red to far red ratio of about 2.89. And as you can see, it's highly enriched in this sort of red to far red region. What we found was when we grew under controlled environments, we were getting a really nice compression comparative to our other glasshouse and bird net sort of external environments. So there was factors of temperature there as well, obviously. We then wanted to drill down a little bit more and look at the difference between, well, if you have enriched in the far red versus sort of this enriched in the blue, what is that doing to time to flower? And what we found was that lentil, faba, and lupin were far more responsive to this far red enrichment. And we were getting quite big differences, as you can see, in time to flower between those two different light quality tests that we did. Now, this is just a snapshot of the work that we did. There was actually, a many series of experiments where we looked at trying to optimize this as best we can. And we really kept coming back for our general purpose industrial kind of scale platforms to this AP67, which is this one here. And we found that to be a really beneficial to the acceleration of flowering time in our species of interest. We also sort of uncovered a few other things that are worth touching on. One is that our developmental responses, such as flowering time, are improved by including absolute photon load in the models. So the more photons that are landing on the plant at a particular light spectral position leads to a different developmental response. So the higher number of photons in the far red region was the best suited for floral induction in this experiment. And if you have a look here, we've drilled down in lupin, and you can find the details of this in a plant cell tissue organ culture publication from 2016, where we looked at the number of photons and the days to flowering in an early versus a late flowering genotype of lupin. Mandalite being the early, Tangel being the late. And we were able to show that the more photons you had, the faster was your time to flowering, as you can see clearly here. And I think it's important to keep that in mind whenever you're setting up a closed environment, growth environment for your plants. Keep in mind that this photon load is also a critical component. We took the opportunity to also drill down into the effect of these far red enriched lights on the underlying gene networks. And for this work, we had some funding to work in subclover, which we'd been working on alongside our cool season grain legumes. Subclover is subterranean clover. It's also a legume species. It's grown on about 18 million hectares here in Australia, so it's a major species for us, and one of the reasons that we use it in a lot of our research work. So when we looked with RNA-seq at what was happening to the gene networks that underlie the different flowering time, we were seeing under far red light versus blue enriched light, and you'll see here the time to flowering differed by about 2 weeks. We were able to show that there was differentially expressed genes related to light signalling, meristem identity, and flowering promotion identified as early as the 3rd leaf stage under that far red enriched light. So, it was switching these genes on really quickly. And you can see the work. We didn't end up publishing this, but it did go up on Biorix, and you can find it there if you're interested in having a look. So what that told us was that, it really happens quickly. It's important to germinate the seed under the far red enriched light. It's also important to think about what happens to plant defense genes, because one of the things that we found in that work was that they were preferentially putting out their reproductive genes and expressing those reproductive genes at the expense, if you like, of defense genes. So if you were looking to phenotype under this far red enriched environment, you'd want to be really thinking hard about whether you were going to get really good results, or whether in fact your pushing of them to their physiological limit under the far red light is going to affect the real world phenotyping results that you might otherwise get from disease, for example. We also showed that the effect of spectral enrichment's really reduced at those lower growth temperatures. So that higher sort of spring light, late spring temperatures for us, which is about 24 degrees day, 20 degrees night, was kind of optimal for the work that we were doing. There's no margin for error. We're growing these things in small pots. You've got to be really on top of your water, your fertilizer. You can't have any pH variability. You really have to be very rigorous with all your phytosanitary regime as well. And like I was saying, they're preferentially diverting resources from defence to flowering, so that has implications for your phenotyping for things like disease. So once we'd sort of worked through, well, yeah, we can use this far red enriched light, these late spring temperatures, these controlled environments to really compress time to flower in our species of interest. We wanted to look at, well, how do we make that now into a platform that we can deliver to industry? What else do we do to make this scalable? And one of those things that we looked at was applying this within a vertical farming situation. So I think everyone should be familiar with what vertical farming looks like. It basically stacks layers within your controlled environments, which maximises your space efficiency. And when you think about how expensive it is to heat and cool those controlled environment spaces, you can really see that it's going to be beneficial to be able to put as many plants as you can per square metre. It also opened opportunities to use each tier of your racking to have a different crop or a different stage-specific optimization of your light. You can tailor the layers to the photothermal needs of each different crop, or of each different stage of what you're looking at with your crops. And in order to really make the best use of this, we had to answer the question, how do we manage the plant height without compromising that cycle turnover time, which we were so focused on, and also looking at keeping a really good, viable seed return. So one of the work one of the sort of areas of work that really underpinned this was working with Sergio Oschat and Federico while he was doing his PhD on looking at applying anti-gibberellins to manage plant height. And this is, you'll find this work published in this place here, so you can easily have a look if you're interested. What we showed was that you could very much reduce the height of your plant, your internode length is reduced without compromise to your flowering time. And we extended this work to a number of species, and here's just as by example is chia, which is a functional food crop here. It originates in South America. It's really responsive to far red light as well. We were able to use anti-gibberellins, as you can see here, at different concentrations to really get a size that was manageable under our vertical farming situation without compromising the seed that we were getting back from the plants. We also applied this later to quinoa, which is another functional food. Importantly for us, we were able to make it work really well in faba bean, and I know that this is probably of interest to some of our European colleagues and Canadian colleagues, and you'll find this work up on Biorix. It's about to go off for publication, but we were able to get good quality seed back from faba bean under these controlled environments, stacked situations with about a 20 centimetre plant height. So, it's a really nice way to increase the number of plants you can put in per square metre. So once we'd sort of worked through, we can make these things flower, we can make them small, we can make them still flower and be small without losing time. We thought, well, let's now tackle this sort of maturation phase. So we were looking for a robust technique to allow us to germinate immature seeds. So we'd done quite a lot of work showing that, we could do this in vitro. We would prefer for a scalable protocol not to have to go into the lab. So what we tried to devise was an in vivo rather than in vitro protocol, so something that kept it out of the lab. So we had a good look at physiological maturity, and again, this is work coming from Federico, and it's published in Plant Growth Regulation, and we were able to pinpoint that we were reaching physiological maturity, in this case in pea, around 16 to 18 days after anthesis. So we knew that if we were to take the seed at about 16 to 18 days after flowering, that we should be able to germinate that with a robust success rate. And we did a lot of work to understand what that looked like in the lab. We did the same thing in our other species of interest. We wanted to really understand, I don't know what's going on there. Sorry, I'm not sure why that's across to the side. Let me just fix that. Don't know what's going on, sorry. Looks like it's moved some of my slide right out of the way here. I'll just try and pull that back in. We wanted to understand what was going on with the hormones that were underlying this physiology of seed maturation. And what we wanted to look at was, if we grow them under the glasshouse, just normal glasshouse under normal ambient light conditions versus in our intensive far red enriched environments, what was happening to the hormone profiles over time. And we were able to show that the auxin profiles were brought forward by up to 8 days under intensive growth conditions. And that happened across the genotypes that we looked at. And there was significantly lower levels of auxin at physiological maturity under the intensive conditions. So the end of morphogenesis, the initiation of that embryo maturation, really occurred between 10 to 12 days after pollination under our intensive conditions. So this sort of underlined the work that we did to then combine all of our pieces together. So we were saying, okay, we know that we can take them to early flowering. We can get them to flower in 25 to 30 days for most of the species that we look like. We can then that we're looking at. We can then compress that seed maturation to about 16 to 18 days, because we're seeing that actually the auxin profiles and the normal hormonal patterns that you see under seed development were being compressed as well. So we could take the seed at 16 to 18 days reliably across most of those cool season legume species. And then we were able to put that under a desiccation for 3 to 5 days, depending on the size of the seed. The bigger the seed, the longer the period you desiccate. And then we were able to germinate those really successfully in soil. So that was part of our process. So that sort of led us to the next question, which was, okay, we've got this protocol that shortens things. How do we integrate phenotyping and start to look for alleles that are beneficial? You know, how do we do that without sacrificing generation turnover time? And I'll just give you a few examples here that we did. So we were able to look at integrating a herbicide screen. So we did this with marker-assisted selection. We were able to run the metribuzin over them. We were able to see which ones survived. We were then able to check for metribuzin tolerance genes, which had been previously identified. And then we were able to quickly cull populations. So the breeder would send us many F2s, and then we'd run these, we'd screen them, we'd look at the markers, and then we'd cull them right down, cycle them to F5, and send them back to the breeders. So that was a sort of a neat way to integrate speed into phenotyping. We also were able to develop a boron and aluminium tolerance screen using hydroponics. So these are each of our lines running in a hydroponic screen. We would give them the toxic levels of either the aluminium or the boron, and then we would look at the root growth. We'd stain it, look at root growth under those conditions. For boron, we could see the leaf margin changes. We'd rate them. We'd then recover them and put them back under our rapid generation cycling. So we were only losing sort of 1 to 2 weeks in one cycle, but we were getting all the phenotyping data that we could then feed back to the breeding programs. And that's work that Richard Bennett really led, and that was published in Plant Methods. So if you're interested, have a look at that. So the final product of that initial work was that we were able to get this 5 to 7 generations a year across a range of species. Vetch is a relatively newcomer. I've added that in because we've done that at SARDI, but the rest of this work was all done with the colleagues at UWA, and further information can be found in these documents here. So we're basically optimizing that spectral and photon load. That's forcing early flowering. We looked at that physiological maturity. We were able to get the precocious germination. We were then able to integrate screening and marker-assisted selection and return the germinated seed back to soil and start the process again. So it's just been a really neat way to kind of turbocharge our breeding programs here in Australia. And this is something that we delivered at commercial scale to the breeding programs until about 2022, when it was then taken into other people's programs and actually integrated in the different areas that it was required within the breeding programs themselves. So a nice real world example of this, that we looked at herbicide tolerance in chickpea. We took the F2 seed into our accelerated single seed descent cycling. We had 500 plus of them in 2 populations, so it was 1,000. We did the trait screening with the markers. We culled the lines without the markers. We sent it back in time after 32 days for field testing. So they did this out of season, and that led us to publish a paper together with the breeding program and other collaborators to show how the integration of your marker-assisted selection into your breeding pipelines allows you to release your cultivar, up to 3 years earlier than if you were to go with a conventional breeding pipeline like we would use for a diploid inbreeding species. And you can find that work in Frontiers in 2022. So nice real world example. I think it was a nice way to wrap up the work that we did, which kind of finished in about 2022, showing that it had this impact in our actual breeding programs. Essentially, in the end, we ran about 50,000 individuals through our ASSD platform for the breeders. We also had a lot of gene discovery from the mapping populations that we were developing for pre-breeding, so salinity, vigour, root lesion nematode resistance, sclerotinia, and there's a number of publications. If you just search for ASSD and gene discovery, you will find a number of populations that we made being reflected in that work across sort of the pre-breeding landscape here in Australia. So then our question was, well, how do we use this ASSD approach to speed our interspecies enrichment? How do we go and collect better variability from our wild relatives? To do that, we knew that we had to synchronise and compress flowering time between wilds and domestic so we could cross them quickly. We had to work out robust hybridisation methods, and we also had to work out really good ways to efficiently clone our F1s to get really big numbers of F2s. And I'll just step you through that. So we looked again at optimising all the factors that we'd already optimised for chickpea, and we looked at the wild relatives this time. We looked at that long day photo period, which was about 18 hours, optimal for our wilds. 24-20 was still the same temperature regime we were using for the domestic, and it worked really well for the wilds. Our far-red enriched light seemed to also trigger good compression of flowering. And obviously we had our optimised fertilising watering as we had previously. I think this work is quite a neat example of how useful this tech can be. So here we have wild cices that were recently collected from the Fertile Crescent region. So that was done in the late sort of 2018, 2019, across that period. And that was brought into Australia through quarantine. They were having trouble getting these things to flower, particularly some of the lines. So they sent them over to us at UWA and said, what can you do with these things? See if you can get them to flower. So we vernalised them. We had limited seed. We had some vernalised, some non-vernalised. And we put them under our far-red enriched light. And we found incredible compression of flowering time, even compared to those that had been vernalised. So we had 25 to 40 day flowering times compared to what was looking like over 100 days when they were putting it through conventional glasshouses. And it also solved this sort of asynchrony that we would have otherwise had, as you can see, between the wilds here and the domestic species. And it allows us to really overlap domestic and wild flowering times. And this is looking at the distribution or the variabilities of days to flowering over 4 of our good cultivars here in Australia and 20 Cicer reticulatum, which are primary gene pool species of Cicer. And you can see that we're getting nice overlap, and we were able to then do crosses quickly and get our F1s back. We were able to confirm those F1s using markers that we developed. We cloned them to bulk F2 seed, and that was a really neat way. You just vegetatively propagate. We needed to get large seed numbers, particularly in the example I'll give next, because there was fertility barriers. And we use this ASSD platform to progress the rills from F2 to F6 in less than 12 months, and we're able to lodge a mapping population from these newly collected Cicer reticulatum germplasm crossed with one of our local cultivars into our gene bank for use by all our pre-breeders, and that remains there as a really good resource. We looked into the secondary gene pool, so going a bit broader, we identified that there were sterility issues, as been widely reported in literature for Cicer echinosternum. We've been continuing to work on this with a PhD student between Judith Lichtenszweig and other collaborators. Alistair Hockey's our student. So we're sort of still digging away at looking at predicting what lines of Cicer echinosternum might be more compatible with our Cicer aretinum domestic species. But we were able to find some in the new collections that had partial fertility. So we knew that if we got big enough numbers, we'd then probably be able to get to a mapping population that would be beneficial to our pre-breeding. So we basically used 2,000. We used that cloning to produce 2,000 F2s. That was reduced as we went from F2 to F5 down to 200. So we've lodged an F5 200 line domestic by Cicer echinosternum secondary gene pool population into the Australian Grains Gene Bank now as well. So that's just a really nice use of the technique to bring together those disparate flowering times to then cycle through quickly once you've got an F2, and to deliver these mapping populations that are really going to be useful when you start to look at what you can do with wild alleles. So we renamed this as the Rapid Allele Mining Platform because we'd included this cloning in there. We also were looking to use the precocious seed germination, and in doing so, we could really deliver these F2 to F6s in less than 12 months, and we continue to do that here at SARDI and at UWA. So what else could we do with it? We were really intrigued by this point, going, well, this is cool. What else can we do? So there was a big investment by GRDC in ascochyta blight in chickpea, and as you can see here, there was a lot of collaboration across a lot of different organisations, including ACADA. We were leading program 3 when I came here to SARDI, which was looking at working with our partners at Agriculture Victoria to do genomic selection and to try and bring together land races, alleles from across the spectrum of the available germplasm that sat here in the gene banks in Australia, to go as broad as we could to search for those minor alleles that may also contribute to ascochyta blight resistance. It was something that was and continues to be a difficult disease here in Australia. We have a different isolate sort of mix than what we're seeing in our overseas collaborators' populations. So it's quite a difficult problem. So working with AgVic, and this is work that's really led by Dr. Sikachuan Kaur and her team, we phenotyped really carefully across field and in pots here at SARDI, and that's work led by Dr. Judith Atieno here. We went back to the gene bank, we got the new germplasm, we phenotyped it carefully, we then genotyped it using a pulse SNP chip that's available at Agriculture Victoria. We developed training populations, genomic predictions, selected our parents, and then we ran them through this rapid cycle breeding platform. So it's a modification of accelerated single seed descent, which meant that we were able to rapidly bring together these complex populations. We had a lot of intercrossing between F1s, and then to deliver the lines back out to the breeding programs, and that's where they're sitting at the moment. So F6 lines, with which we've shown through another round of phenotyping to be highly resistant to ascochyta blight, are now with our breeding program. Touching on the tratomic work that I alluded to in the first sort of minutes of the presentation, what we're doing in oats is to take a reverse genetics approach. So I'm not sure if everyone's familiar with the tratomic platform, but I'm sure many of you have heard of it. They use a find-it approach, which they published recently, to identify targeted point mutation candidate genes. So you supply them with the candidate genes. They then look through their libraries, and they came here in 2023-24 and developed a big oat library, which we're now delving into. So fortunately, the material is here in Australia. We don't have to bring it through quarantine. It's a hexaploid, so there's mutation across the different homologs. We're using this sort of rapid allele mining approach to rapidly bring the mutant lines that are coming back from tratomics together to cross the mutants by mutants to develop these double and triple mutants that we will then test. In this case, we're trying to reduce the oil content of oat to improve its shelf life, improve its mellability. So what we're hoping to do is to do dosage knockout, basically, of the different genes that we think are important in that oil deposition pathway. And we're going to be using the accelerated approach to bring together those to test our hypotheses really quickly. In oats, we can get a 2-month seed-to-seed cycle as well under the similar sort of approach as what we've done with the cool season legumes. Just a quick touch on pasture legumes. I'm coming to the end of my presentation now. We've used a combination of physiological seed dormancy breaking techniques and an accelerated SSD approach to actually develop a new cultivar that's powdery mildew resistant. This was done very, very quickly, and we were able to do the breeding component in 2.25 years instead of 4.5. And we, at 6.5 years, the 6.5 year point, we've been able to put this into commercial seed evaluation with the breeding company partners there. So this is a really nice example of how you can use it to just speed up your breeding. Pastures were genuinely, laggards when it came to breeding technologies, and they were taking a really long time to breed. So it's a nice incorporation of both the interspecific parts, because this is a cross between a strand and a barometric to bring across the powdery mildew resistance. And then it's also this acceleration within your traditional breeding to get back to homozygosity. And I think finally, we'll just talk about wheat. So we had a really nice recombinant inbred population of wheat here. I was saying 200 lines there, but they started with 2,500 lines of wheat, which they've kind of reduced down to around 200 by tossing out the ones that have got problems with, photoperiodism and vernalization and other things that have come across from a wild parent. So this is, again, an introgression between 3 species. We've selected lines that we really wanted to put out into the field, but we had the problem that because this was a common inbred line mapping population, there just weren't high seed numbers. So we looked into the literature, and this is work led by Dr. Yusuf Genk, realised that the determination of how many seed you're going to get happens really early on, and this is work coming out of University of Adelaide, Dr. Scott Bowden, showing that the spikelet is there at that 3-leaf stage. So we did a combination approach of putting them under ideal conditions until that 3-leaf stage, ideal conditions for wheat being, a shorter photo period, cooler temperatures, and then bringing them out into the glasshouse under the lights. And with 3 months, we had up to 7 tillers per plant, heaps of seed, and it's a really neat way to then turbocharge your wheat breeding as well. So that was a nice, a nice add-on to the work that we'd been doing. So to finish, our rapid generation cycling enables faster introgression of alleles of value. It works across a range of species, that's I hope I've shown, and in a relatively genotypically insensitive way. Obviously, light quality is important. It's secondary to light duration and to temperature, of course, but it has a role to play in near synchronization of flower in those phenologically divergent genotypes. It's important to have a look at each of the genotypes and particularly the species that you want to look at. What we've found is that within families, some are more responsive than others to that far red enrichment. It's not going to work for everybody, but do consider it when you're developing these sort of platforms. And I think that the applications are only bound by the imagination. I've come here to SARDI and I've seen this work sort of creep into areas that I wouldn't have thought of 3 to 5 years ago. And I hope that that you've got some ideas of your own that you'll bring to this kind of area. So there's a huge amount of collaboration that's gone into this work that's just touching on some of the collaborations that are existing. And on that, I'd like to ask if there's any questions. That's a photo of our family farm in Victoria. We've got a nice outlook to the Grampians, and that was taken in a good year.
Thank you, Janine, for your presentation. It was full of very interesting results and technical aspects too. So let's see if there's any question from the chat or from the people. You can unmute your mic or raise your hand. Okay, so there's Tom raising his hand.
Hi, Janine. Thanks for a wonderful talk. Nice to see you again and hear you again. See you. Yeah. I guess maybe a little bit philosophical question. When you've worked with plant breeders and talking about accelerated SSD, do breeders use the approach, let's say, for all their crosses, or it's more of a technique for special crosses? And we can define special in different ways. Just wonder if you have a comment about that.
Yeah, certainly when we were delivering this for all of the pulse breeding programs, they were sending us their special crosses, the ones that they were really interested in incorporating into their pipelines as quickly as possible, which, makes sense. And I think that example that we published in 2022 with Christy Hobson, who does the chickpea breeding here, you know her well, is that they were really interested in that herbicide tolerance trait and incorporating that as quickly as they possibly could. So that one came to us. And it also came here to SARDI, who we were working in collaboration with at that time, to look at the markers. So, that was just a nice confluence, I think, of special things and using all the tools she had at her disposal to get as quickly as she could to the outcome she wanted. So definitely special things. A lot of mapping populations were developed by us for the pre-breeding programs. And I think that's where we got a lot of value in terms of gene discovery. So it was definitely biased towards the important key traits that they're after. I would say, though, that with the work that we've been doing here at SARDI, we can do this in really big glass houses. You can just fill out your glass houses with LED arrays as well and get, almost as quick. You don't have to do it in specific controlled environments with really tight regulation of temperature. So I know that they're doing that now. They've adopted these sort of protocols in our quarantine facilities. There's a lot of the breeding programs across Australia have put them in, and it's really nice to see the work get that kind of reach. And I think as it gets that reach, it becomes less specialised, and people might be starting to go, well, we'll just use this as a routine approach.
Thanks a lot. Thank you.
No worries.
There's a question from Paolo.
Hello, Janine. Thank you very much also on my side. I'd like to ask a couple of practical questions. First, is there an ideal, near ideal kind of pot that you use for plants? Because sometimes I guess that you would like to fit a lot of plants into the equipment, so this might be important. And secondly, how do you water the plants? Is there like a micro irrigation system or just something from up? I mean, something very simple, or, because perhaps it's also a little bit a delicate issue.
So we've got some nice detail around pot size and soil types and those sort of things in the publications that are listed there. We vary the pot size by the root architecture. So for branching roots, we can go with a squat, small, sort of 750 milliliter pot. Square one is best for us. For the more taproot species or the longer rooted species like lupin, we went with a taller 1 litre pot. But certainly you can cram a lot of that size pot into your space, which was very useful for us. We were pedantic, Paolo. We had hand watering because we wanted to see the plants every day. And we found that when we tried to do watering through, drippers or anything else, it just wasn't working for us. The plants were getting more disease because they were getting too wet for too long, or they were getting mist and drying out and disappearing from our system. We were just more comfortable at UWA doing it with a very specific pot, individual pot watering. And we had really good skilled technical support. And, those guys were on the first slide. They were amazing. We couldn't do this work without them. I would say that working on canola, you can flood irrigate. Working in the cereals, they're a lot more robust. We just put them in big trays, the cereals, and that's similar to the work that you see coming out of the speed breeding publications of Hickey's group at UQ. We put them into just trays, and then we sit the tray in another tray. So as their roots develop, they've got a bit more space, and then we just water them en masse. It's not as detailed as we were doing previously, because on a pot by pot basis for watering, we were also doing pot by pot basis for fertilizing, and that was happening with a known amount of fertilizer every 2 weeks. So when I talk about these regimes that we adopted, they were quite stringent.
Thanks.
Thanks, Janine. Are there any other questions from the public? Okay, so it appears that this concludes our presentation. Thanks again, Janine. It was very interesting. The presentation will go on our website very soon. I remind everybody that there was a link in the chat for giving us feedback on the presentation and all the technical aspect and how to do our job better. And I also put the link for the next conference ILS5 that is starting to pick up momentum. Thanks again, Janine. It was a pleasure to hear your contribution.
Thanks so much, Nelson, and it's nice to see the others there. So yeah, nice to see you, Tom, particularly.
Bye.
Thank you.